Welcome to MOOC NPTEL course on Bioengineering, an interface with biology and medicine. Let's kind of first start with some basics, some terminologies. When we say genotype, it means all the genetic contents of a given individual, all those genetic information, and phenotype, how actual individual looks like, their morphology, their phenotype characteristics. Now let's kind of discuss this part in context of Mendel's experiment. So Gregor Mendel, he was a scientist who did genetics experiment on the pea plant, on the garden pea. And while he was looking at pea plants, he observed there are many contrasting characteristics present in the pea plant. And he thought that those are some sort of characters which transmit from one to next generation. And for the same, now we say those heritable factors as a gene. So for the same factors, there might be different alleles which are present there. So one gene might be having different allele forms. So let's say this is a plant, a flower which is shown here is a purple colored flower. Now if a gene codes for it, which is having dominant in trace, so let's say capital P. Now a recessive form of that will be a small p, right? So first I want to make you familiar with the terminologies. Dominant, recessive, homozygous, heterozygous. So let's kind of do one Punnett square cross and try to understand these terminologies. By the way, in genetics, it will be a good idea that you can open your own notebooks, and as we go along, there will be certain problems for you to also try out. It looks very easy, but when you do it, you'll be more confident. So both from the sperm and the ova, from the male and female parts, let's say now you have these gametes. So it is very straightforward, but let me kind of now ask you some terminologies here. So let's ask how many homozygous recessive are here? Homozygous recessive. Somebody rightly mentioned one. So a small p is the recessive characteristic, right? Dominant is the capital P. So how many we have homozygous dominant here? One, capital P, capital P. How many are heterozygotes? Which are 50%, right? So 50% are heterozygous. Now what will be the genotype here? Genotype means genetic contents of this.
One is to 2:1.
Everybody agrees to him? And what will be phenotype?
3:1.
Why 3? Because one is dominant, right? P, capital P is dominant. So I'm sure now you already know yourself all the terminologies. So in this case here, the genotypic ratio will become 1:2:1, and phenotypic ratio, all those which are dominant, which is purple in color, is the phenotypic ratio as compared to the white, which is recessive color, right? So this is how I think it's a good idea for us to learn the terminologies, and this is what Mendel was looking at it. But before I come to Mendel's experiment, I thought to make you familiar with the properties and these terminologies. So in this case, we got 3 purple and 1 white. Ratio was 3:1, which is phenotypic ratio, and we have 1:2:1, which is the genotypic ratio. Again, let's kind of go back and think about it at the cell level. We have already talked about it. And from the individuals, we have billions of cells. Each cell contains those nucleus. Within those, we have the DNA contents, chromosome. From those, we have the genes. What Mendel was thinking that these genes, what we think now, is the sequence of nucleotides at a specific place which contains some of the heritable factors, which contains some information which could inherit from one to next generation. Later on, people realized that this could be termed as a gene. So now what we think is gene is what Mendel already thought that time as the heritable factors which can transmit from one to the next generation. And there could be different alleles of a given gene which are having one copy from each parent. So in this case, let's say we say the capital P and small p, right? Those are for the same color characteristic, purple or the white. So these are the two alleles of a given gene which are inherited from the parents. So as you can see here, the alternative variations of these genes are known as alleles. For purple flower on this chromosome or the white flower here, these are the pair of homologous chromosomes. So each organism harbors two copies of a given gene. A dominant allele has the effect, but recessive does not show an effect. And that's your dominant, it is known as dominant characteristic, which is going to show its properties. Now in this context of purple versus white color, if you look at this sequence of the gene which codes for the purple color versus the sequence of this gene which is for the white color, it is exactly same, just one base pair change from C to A. That's the only change here. But because of that one change, so when we talk about the sequences, we are talking about only some letters of ATGCs. They have to be in a defined manner. And sometimes just by that one change of one base can result into lot of misregulation, lot of deformities. And many times in the cancer patients, people find out lot of mutations happens. Sometimes something gets shift, some base pair shifts, and therefore the entire protein coding sequences can be changed. So in this case, it's very interesting example here. You can see that because of one base pair change, this is going to make enzyme which is required to give the purple color, whereas this one has deficiency, it cannot make the enzyme for purple color, it only shows the white pattern. So same gene sequence just with one base pair change. This is why these are alleles of that given gene, and it is going to show you the two different properties of purple versus white color. So now let's come and talk about Mendel, what he did, and his experiment on the inheritance concept. So Mendel was doing experiments not for a job, not for the sake of a profession. He was doing these things out of his interest, and he was actually a monk who was studying the pea plant by growing them in his own backyard. And at the age of 21, he joined this Augustinian monastery. But he did not like that too much, and then he felt that he should do more advanced study in physics and chemistry. So in fact, he was much more interested to study physics, chemistry, maths, not biology. Sometime you may not like a subject, but you might be able to still make an original contribution to the field that you will be remembered for that contribution. So after he returned back from the university, then he again joined that monastery and started teaching in a local school. And he was just growing the garden peas in the garden, and then he was making a very close observation, very meticulous notes of what is happening from one generation to the next generation. So each time when he do a cross, he will count that what is the properties he's looking at, how many seeds he's growing, how many pea plants made out of it, what are their each characteristics. And because he was doing it so meticulously, he was able to come with certain mathematical numbers. And he was very astonished himself that there are many properties which are showing similar kind of patterns. So then he was able to formulate certain rules and certain laws, which became Mendelian laws of genetics. So he chose only true breeding varieties. Now, does it make any sense, what is the true breeding variety and why he need to choose the true breeding variety? So Mendel chose only true breeding varieties, is a statement for his experiments which he did on the pea plants. What could be true breeding means? Somebody tried saying that no mutation. Any better explanation? Homozygous. So So you are ensuring that if it was purple color coming, purple color can come out of either, it can be capital P small p or capital P capital P. So you are ensuring that they are going to contain the same characteristics from one to next generation. So therefore he was choosing those characteristics and those plants which are true breeding. So in 1860 he was able to come up with the law of inheritance to define the genetic principles. So what are these heritable traits? These are heritable traits are many characteristics, like sometimes our eye colors, sometimes hair, different type of hair patterns are there, different type of eye colors, yes.
But how exactly do you choose true breeding varieties?
So ideally, if you are crossing a purple colored one with purple colored one, and you are still getting the progeny is purple colored, you are ensuring that you are doing one to next generation those crosses, and still you are getting purple colored. They are not segregating. It means those are true breeding. Does that answer to your question as well? So what are the genetic principles which accounts for the transmission of these traits? Well, many of the rules which were deciphered by Mendel as the hereditary rules. So I'm sure you can observe different patterns of hair, different colors, different type of eye colors. All of these are natural variations which are there and they still keep passing from one to the next generations, and we do see that there are heritable characters, heritable variations. So how to define hereditary? People have tried to come up with different hypotheses that how to define hereditary. One hypothesis is known as particulate hypothesis, which is essentially you are playing the cards, you are shuffling the cards, and while shuffling the cards from while distribution, it is always random, right? You have no idea that how these cards are going to be distributed to different players, from one to second to third player. Genes are like that, from one to second to the third generation, probably you are shuffling the cards, and that is known as particulate hypothesis, which became more popular. So here, genes can be shuffled and passed along from the one to next generation. Another hypothesis was blending hypothesis. It means from the two parents, genes are coming and they are blended like paint. So like paint color, and they are going to give rise to a different color, like from the blue and the yellow paints, if they get blended, they will make the green color. So this hypothesis is not as popular, but these are again some hypotheses to explain how to define hereditary. So Mendel chose pea plant, of course because of availability of pea in the garden, what he was, where he was in that monastery. He felt that these are the experimental system which can be done very fast, very short generation timing, and it has many characteristics, at least 7 he observed, which are derived looking from the same gene having different allelic forms. Then you can generate large number of seeds and you can make those countings. In any of the biological experiment, statistics become very important because you have to ensure that you have enough N to prove or disprove your hypothesis. And that's what was again good system here, because if you think about doing experiments in the animal system, if you'd have chosen mouse or any other animal model, then it may not have been so easy for him to grow that many number of model system to do statistics, and you cannot grow them so fast either. He was also having full control about doing the self-pollination or the cross-pollination of the pea plants for the breeding purpose. So as a result, all of these things were quite helpful for Mendel to think about doing genetic experiments. As we talked, he chose only true breeding varieties. He was crossing them to multiple generations, only selecting those which are showing the same pattern. And he selected only one characteristics at a time for his studies, which are having distinctive alternative forms. So for example, purple versus white color of the flower. That is one characteristic he tried to study. We already talked about some part of that. Flower positions, whether it's axial or terminal. Seed color, is it yellow color or green color seed? Seed shape, it can be round or wrinkle shaped. Pod shape, it can be inflated or constricted. Pod color, green or yellow. Or stem length, tall or dwarf. So many people say that Mendel was actually very lucky because in the same system he was able to get for the same gene, different allelic forms and many properties, which usually not present in many other plants or many other animal systems. So he was pretty lucky to randomly selected pea plant as the experimental system, and he did all the experiment which became very reproducible, and now we have very strong foundation for those laws. So these characteristic variants are called traits. So to elucidate principles of heredity, inheritance of each trait is determined by the units or the factors which we now know as a gene, which are passed on to descendants which remain unchanged. Individuals inherit one such unit from each parent, and then they transmit from the one to the next generation. So now brings to the Mendel's first law, which is law of segregation. So just to kind of brief you, this is the stamen or the male part of the this flower, pollen producing organ. This is the carpel or the female part, which is egg bearing part of the flower. So for doing this experiment, he was ensuring that for doing the crosses, it has to be very well under the control condition, just so that there is no cross pollination from one to other, different flower and different characteristics. Whatever he wanted to study, only those pollination he wanted to do that. So he cross pollinated only 2 contrasting and the true breeding pea varieties. So purple versus white, for example, or long versus short, or looking at the yellow versus green, seed shape, etc. So his doubt and question is that what is it dominant or recessive we are looking at over there or not. So all you want to ensure here that you are only studying the homozygous kind of characteristics, because capital P or capital P small p, both are possibility for purple color. But can you choose only capital P and keep growing them further for your experiment, because then only that you are only talking about capital P versus small p. So then you have the good experiment to be done.
Plants were true variety, true breeding. How do you make sure that the peas work?
Right. So lot of interesting ideas, of course. How he was ensuring these things, as I showed you, he was doing these pollination experiments in a very controlled condition, so that there is no cross pollination happens from the neighboring plants which can influence these characteristics. So if he is only growing the purple colored one and then he is growing white colored one separately, then only he will do the cross of those two to ensure that only the true breeding varieties cross can happen. So hybridization is the phenomenon which is mating or the crossing of 2 true breeding varieties, and this is known as the F1 generation or the filial generation. Just get familiar with the terminology. P means parental generations, parents. F1 is the filial or the progenies. So in the first, when he did the cross, the first generation looked like all purple colored. So now let's talk about his crossing experiment, where he crossed purple versus white, 2 of the contrasting traits. So this is dominant color trait, the purple colored, versus white is a recessive trait. Now P generation or the parent generations, these are true breeding parents, purple or white. F1, all of them showed purple pattern, and when he again crossed them further, it distributed into the purple versus white, 3:1 kind of ratio. And he did this experiment on many of the characteristics, not only the purple and white, among all of the contrasting characteristics of different P properties which he was studying, and therefore he was able to come up with some conclusion that looks like they distribute in a very specific ratio. It is not very random phenomenon. So the F2 generation is when he is doing the F1 hybrid, self-pollinating them, and then they are resulting into large number of these seeds which he was growing further, and then able to come up with these numbers. So again, what you can see, there are large number of flowers which he was counting. So 700 purple flower with 220 or so of white flower. So this is what then he felt, that these are the allelic forms of a given gene or factors which are showing the same property, but only one contrasting difference is found. This is how the cross will look like. So we already briefly talked about the Punnett square. So if this was the cross, the purple flower versus white flower, these are the gametes, capital P and small p will be derived. In the F1 generation, you will have this hybrid, capital P, small p. From the male and the female parts, the egg and the sperm, you are doing this cross, and that results into this ratio of 3:1 of the phenotype. As I mentioned, he did this on many properties, not only the purple and white, also on the flower position. And the ratio was not exactly 3:1. It was 3.15 or 3.14:1. But he was finding similar kind of patterns, similar numbers in many properties. So the ratio of dominant versus recessive trait was 3:1. Now question is, if we have one purple flower, how are we going to determine the genotype, whether the purple flower is capital P or capital P small p, right? If we have purple flower, it can be two properties possible. So let's imagine two situations. One is capital P and second is capital P small p. Both of them are purple flower. If I give you some seeds and I tell you that all these seeds are going to make the flowers which will look like purple, can you determine the genotype or their genetic makeup? Are these capital P or capital P small p? So Mendel was doing this experiment himself. He was doing a lot of back crosses, and one of the cross which he observed, so please do try yourself as well. Whatever I do, you should try it out yourself. So we have two possibilities. One is capital P, second is capital P and small p. He made a cross of these two conditions with homozygous recessive, which was small p. With this cross, now if you derive yourself, so what kind of numbers can you get now in terms of ratios? In the second possibility, what is the ratio?
1:1.
So, yeah, so you have 1:1 or 50% here, because these are going to show you the purple colored flowers, these are going to show you white colored flower. So if the genotype or the genetic composition of that was capital P small p, then you will see 1:1 ratio. If they were all going to show you purple colored flower pattern, it means genotype of that seed was capital P. So this is how this kind of test can be done, which is known as test cross. And test cross, keep in mind, you are doing always with the homozygous recessive. Test cross always done with the homozygous recessive characteristic. So in this case, with the two crosses which we just talked, you have possibility of capital P or capital P small p, and you are crossing with the homozygous recessive characteristic of small p. Either all offsprings were purple colored, or 50% of them were purple and 50% of them were white colored. I think it's good to be more attentive in genetics lecture. It's not only interesting, but also a lot of questions are going to come based on that for sure. And as long as you know how to make these crosses, Punnett squares, you can very easily do these kind of questions. So please make sure that you are trying all of these things yourself. Whenever we say that make this cross, do try out yourself. Looks very easy sometimes, but still try to do that. So breeding an organism of unknown genotype with a recessive homozygote is known as test cross, which can reveal the genotype or the genetic composition of that particular organism. So what we can conclude from this section so far, that the law of segregation suggests that genes have alternative forms or the alleles. There are two alleles for a given trait which is heritable, and those can get separated during the gamete formation. It also explained that the ratio is approximate 3:1, and a trait may not show up in the individual, but you can still obtain that information from the test cross, which actually can be passed from one to the next generation. That brings us to the second law of Mendel, which is law of independent assortment. Mendel, when he was doing the experiments, of course he did not make the law that time. So he was doing the experiments, he was making those observations. After looking at many experiments giving the same ratios, then only he was able to come up with certain hypotheses that this particular thing is working well, and now this can become a rule. So in terms of looking at this now, he started thinking about, earlier we studied only one characteristic, one gene and two allelic forms of that. Can we now start looking at two genes, two different properties, different allelic forms? So for example, seed color and seed shape. This is a round seed and yellow color. So we are looking at two properties now, right? And that is known as dihybrid. When we are talking about studying two traits simultaneously, that is known as the dihybrids. Or you are talking about a seed which is green in color, but either it is round or it can be wrinkled. So you are looking at two different properties here. And to denote them, you can say it is capital Y, capital R, or small y, small r, small r. So although I mentioned to you already that Mendel derived his law as a law of independent assortment, but let's assume the possibilities that what could have been either dependent or independent. So for doing this experiment, from the gametes from the parent generations resulted into F1 generation, which is this hybrid, capital Y, capital R, small r. So now let's assume that Mendel was not sure that what should be hypothesis. Are the two properties which we are studying at the same time, they are dependent or they are independent? I'm sure by now you know the answer, it is independent, but let's say that he was not sure that what should be the property. So from this seed, which is the hybrid, now we have capital Y, capital R, small r. There can be two possibilities. Either the gametes which are derived are having the capital Y, capital R, small y, small r. Only this kind of characteristics are getting distributed. Or it can have multiple ways of distribution. Capital Y, capital R, small y, small r, and additionally we have capital Y, small r, and small y, capital R. So there are two possibilities now, right? Can you please start doing the cross yourself and tell me that what will be the answer from these crosses? So Mendel's law were actually based on the result which he obtained. But hypothesis is something, when you are making hypothesis, you have no idea that what the results will be. So at that time you are essentially just exploring all possibilities. It could be multiple characteristics might be depending on each other, or they might be independent of each other. Both are possibilities, right? So if these are the characteristics, so now how best to do the crosses and derive the conclusion out of it? If you do the crosses, what will be, if this is the gametes, then what will be your progeny look like? Please do that fast. You have to derive all the, no, this is not correct. For this one you have to have all of these gametes, because from the male and female both you have to derive. So I think looking at things sometimes looks much easier, but when you start doing yourself, then you realize that you can make mistakes easily, right? Alright, so just to save time, if you do this particular cross here, so male and female gametes which you have capital Y, capital R, and small y, small r, if that was the case, then the ratio could have been 3:1. But what he found was something different. He found the ratio as 9:3:3:1. So the round yellow, round green, wrinkled yellow, and wrinkled green, they are showing phenotypic ratio as 9:3:3:1, which was different observation than what he was actually assuming. If the characteristics were depending on each other, the ratio could have been 3:1. But because they were independent, they are sorted independently, then the ratio observed was 9:3:3:1. So each pair of alleles segregate independent of other pair of alleles during the gamete formation. So from this section we can conclude that the second law of inheritance of Mendel was observed based on the studying two characteristics at a time, and he stated that two or more genes, they can be assorted independently, and each pair of these alleles segregate independently during the gamete formation. After crossing the experiment, then he observed the ratio, which is 9:3:3:1. So Mendel, when he proposed these laws and brought those calculations, those numbers, he published in a small journal, and that time, as I mentioned, he was not doing these things for just doing research for and science from publication point of view. He was doing these things out of his own curiosity, and he was not biologist per se. He was just doing these observations based on his interest of observing the nature and the plants. So he was not very popular. Nobody knew about his findings. He published already his result in 1866, but people were not aware of what Mendel did, and it just got published in some journal which nobody noticed much about it. Many scientists over the period in 19th century, independently they are working on different genetic laws. Also they were looking at Drosophila or the fruit fly as a model system to study the inheritance. And interestingly, they were coming with the conclusions which were exactly same what Mendel already made several years ago. So these three scientists, Hugo de Vries, Erich Schimmark and Karl Correns, they made the same conclusions what Mendel already made in 1860s. So then Mendel was kind of, people started recognizing his contribution, and in many ways the Mendelian laws were rediscovered, because now people have much more confidence that what Mendel has proposed was very accurate, and three independent scientists have confirmed their findings, confirmed his findings. So now everybody started recognizing and appreciating Mendel's contribution, and eventually now he is recognized as the father of genetics. So as I mentioned, let's be open to various observation, the things which we like to do it, and irrespective of which discipline you study, because you never know your training in one area, but your observation towards your interest can give rise to certain different output, and those will be much more fundamental, much more original contributions for any given field. So you are now familiar with the two main Mendelian laws of segregation and independent assortment. In the next lecture, we will discuss some of the examples of Mendelian genetics. You will see how many human traits follow Mendelian pattern of inheritance, and how inheritance patterns are often more complex than predicted by simple Mendelian genetics. Thank you.